All right, ladies and gentlemen, let's get started with today's biology review. And so I am actually just opening this meeting. Hopefully, there you go. Today, we're going to talk about active transport. And today's video should be relatively short. Um, just starting to get my Zoom set up here. And we're going to be talking about active transport. Now, in the previous video, we discussed passive transport, and we said that passive transport was pretty much the movement of molecules across the membrane without using energy. And today, we're going to talk about active transport. And just like the word suggests, active transport is the opposite. Active transport is actually the use of energy to transport molecules across the membrane. Okay, so passive transport is important because passive transport lets a lot of molecules like oxygen, which is very needed, and carbon dioxide, which is also very needed, to pass freely through the molecules, and they move in favor of a concentration gradient. Active transport is kind of like the opposite. So I was explaining where I was trying to illustrate that active transport, we can kind of visualize it as this. Passive transport is kind of like moving in favor of the water, going with the flow, so to speak, right? And here, whatever vessel, or it just moves freely. Now, passive transport, or this is passive transport. Now, active transport would be the opposite. It would be moving against the concentration gradient. So you would probably have the same object just moving in the other direction. And this one requires energy. Now, I'm just drawing these little arrows to kind of indicate that I would be moving against the concentration gradient. But, it's nothing too hard to understand. So let's just go ahead and start defining what active transport is. Okay, so according to your textbooks, active transport uh, drives molecules. Active transport drives molecules across membrane from a region of lower concentration to a region of higher concentration. Concentration meaning that basically it's moving against the current, right? So you would require um, the use of chemical energy. So that's something else worth noting, and it's that we uses it use this process uses transport protein. This process uses transport proteins that require the use of chemical energy. Right? So that's pretty much it. Let me. Okay. So let's move on real quick. And one other very important thing that's worth mentioning is that all transport protein span the membrane, and most change shape. Okay, so all transport proteins span the membrane and most change shape most change shape when they bind to a target molecule. Basically, in active transport proteins have to change shape. If they don't change shape, what's going to happen is that it won't function correctly. Remember that one of the principles of biology, specifically molecular biology, is that the shape or the correct conformation or shape actually affects a protein's functions. So all transport proteins have to change shape. Now, that being said, let's talk real quick about um, some more key um, sorry, more key facts on active transport. Okay. Again, active transport. Active transport. Um, or active transport proteins. I'm sorry. Use chemical energy, which we already said, to move across the concentration gradient. But here's the other thing: is that most use energy from a molecule called ATP. Called ATP. And basically, ATP is kind of like that chemical. And so we've talked about we've mentioned chemical energy basically for the last 2 or 3 chapters. But to be more specific, ATP is that energy, or ATP is that chemical energy that cells use. And we're going to talk about ATP in further chapter, but there's just so you guys have an idea, that's the chemical molecule that is used. Okay, now it's used either directly or indirectly, and one of the functions, or one example of active transport, is the exchange of sodium and potassium, of sodium and potassium used in the transmission of nervous impulses. So this is an example of active transport. Okay, in our neurons, we need this to help us function uh, properly. Okay. Another one is the proton pump. Another example of active transport is the proton pump. See if I can write it here. Another example of active transport is the proton pump. And this removes hydrogen molecules, hydrogen atoms from inside the cells. So again, it's very important, or the The sodium and ion pumps, which is also referred to that, are very important in the generating of nervous impulses. And I think we were mentioning in class the example of moving your big toe. That's a series of electrical impulses that go from your brain all the way down to all the nerves, and they exchange or turn that electrical energy into chemical energy. And of course, when we talk about the hydrogen pump, removing the excess amount of hydrogen, because the more hydrogen inside the cell, that means the more acidic the environment becomes, and that can be kind of dangerous. All right, so let's go ahead and delete this, and then let's continue to talk about some of the concepts of all right, let's move on. So let's talk a little bit about endocytosis, and we're gonna define what endocytosis is. Okay, and endocytosis is the process of taking liquids or fairly large molecules into a cell by engulfing them into the membrane. So endocytosis is the process is the process of I'm sorry of engulfing. Endocytosis is the process of, excuse me, of taking liquids or fairly large molecule. I'm sorry, of taking liquids or fairly large molecules into a cell by engulfing them into the membrane. Into a cell by engulfing them in a membrane. Engulfing them in a membrane. So basically, endocytosis is that process by which um, is the process by which molecules are taken into the cell. It's literally like ingesting molecules in more and mainly in liquids or very large molecules. Okay, so basically what happens is that they they reach the receptor, and see if I can illustrate with that what happens here. Very, very simple. We have a membrane. The membrane has a series of receptor proteins, and basically when a real large molecule gets into the membrane or comes in contact with these receptors, what happens then is that the membrane begins to kind of fold inward, and that molecule will enter molecule or fluids, right? Because it says larger fluid molecules. And finally, what happens is that as it folds inward, then the membrane, and this is a very rough explanation, very missing out a lot of the details, but just so you guys have a general idea of what endocytosis is, this is what happens, more or less. The molecule reaches the protein receptor, that then it begins to fold inward until it is actually inside of the cell. And that's basically the process of endocytosis, right? So moving on in your textbook, um, within endocytosis, a specific type of endocytosis is what we call as phagocytosis. Okay, and let's define phagocytosis. Phagocytosis Phagocytosis is a type of endocytosis in which type of endocytosis in which the cell membrane engulfs large particles. Engulfs large particles. So, one second, let's see if I can move this. Basically, and this is very important, plays a key role in the immune system. We have molecules called macrophages or macrophages. I'm sorry, and these macrophages, what they do is they basically eat up bacteria and destroys them. Okay, so basically, in defending, of course, against bacteria and other pathogens. Okay, so basically, um, right? Phagocytosis is a type of endocytosis that helps that literally engulfs really large particles, and they play a key role in our immune system in defending against bacteria and other types of pathogens that can cause diseases. Right. So then let's move on to exocytosis. All right, and exocytosis, and this would be the last concept of today's class. Today's video was lesson is short. Exocytosis. Exocytosis is the opposite of endocytosis. Endocytosis, and basically is the release of substances out of a cell. By fusion of vesicles with the membrane. So basically the opposite of a membrane. So the opposite. And let me see if I can do one of my very famous rough drawings so you guys understand what I'm trying to say. So here's the membrane. Give me 1 second. All right, there we go. So here's the membrane. And we have a particle that is inside of the membrane, and we want to remove it. We want to get it out of the cell. So what happens is kind of like the opposite. It's kind of like gonna spit it out. So again, important to note that the membrane begins to fold, and or basically the particle now pushes it upward on the membrane. So what's happening is that it's pushing upward against the membrane, and it fuses, and then the particle is released. The particle is then released out of the cell. Now, this is very important in the transmission of nervous impulses again, because as I mentioned, let's see if I can try to illustrate it better. Basically, when there is a nervous impulse in our brains, what happens is that, first of all, our brain has a series of a vast collection of, and this is my very rough drawing of a brain, but what I want to illustrate is that there are a lot of nerves and neurons. There is a plethora of connection with neurons, and they branch out here, and they branch out here, and they branch out here and here, and they branch out here, and then this branch has another branch, and there's millions of them. And of course, I'm not going to have the opportunity to illustrate everything, but basically, when you want to move, let's say you want to get up out of bed in the morning, right? Well, there's a message that's sent to the brain, and the brain says, all right, it's time to wake up. Although it's some for ease for others than it is, it's easier for others than for some. Like for me, for example, it's not that easy. Anyway, the impulse, and let me see if I can use a different color to mark what I want. The impulse is sent out here, and there's an electrical impulse which is sent to this nerve for whatever, just to select, which branches out into this one and branches out, and it essentially leaves our brain. Now, exocytosis, what happens is that these electrical impulses have to turn into chemical impulses. So as they travel from one neuron to the other, they are liberating, the neurons, which are nerve cells, are liberating that electrical impulse into another one until it reaches the desired area, and then the movement occurs. So exocytosis is also used in the transmission of nervous impulse. So again, endocytosis when active. So real quick to summarize, active transport requires the usage of energy and transport proteins to go against the concentration gradient, no matter how high the concentration is. Um, we have endocytosis, which is the process by which a cell, or excuse me, in transport, in active transport, we have transport proteins that help transmit electrical impulses with the exchange of sodium and potassium, which also helps with the with the proton pump to eliminate hydrogen. Then we have endocytosis, which is the process by which molecules enter the membrane using energy. We have phagocytosis, which is basically used to or plays an important role in our immune system. And finally, we talked about exocytosis, which is when substances or molecules leave the cell. And an example of that is the transmission of nervous impulse. So guys, hopefully this video will help you out in understanding this a little better. And we will see each other in our next class. So stay safe. Bye.